The next step is to dilute the sample through serial dilution. Okay, so whenever you do any kind of dilution, you're always diluting the culture, whatever the microbes are, into a liquid.
Okay.
So in this case, I'm going to use PBS, which is just buffered saline, just like a salt solution, and that helps the cells like stay intact and not burst when they get added to it, in addition to like the culture. So these tubes, they're 10 to the 0 solution, so it's basically just pure culture. And so I'm gonna start there and take, I'm gonna do 10-fold dilutions all the way across. So I'm gonna add 900 microliters of my PBS into all of these tubes, and then, as I dilute it down, add 100 microliters.
And at the end of the serial dilution, how dilute will the sample be?
So we did 10 to the minus 5, so it's going to be more than 1,000 times more dilute than the initial sample. So it's very dilute. And like the purpose is to get, when you plate it, the purpose is to get as many single colonies on there, and single colonies are formed by one single cell that's found its home on the plate. So that's why we're diluting it so much, so that in this last sample you have single cells floating around, not clumped, and once you plate them, you can get little single colonies on the plate.
And if you didn't, essentially we would just have so many colonies on the plate because there's still so many cells within that not diluted sample.
Yeah, you would just get like a film, and you wouldn't be able to quantify how many cells are on there, which is one of the purposes of doing a serial dilution. Okay, so now we've done our serial dilutions and we're ready to isolate single colonies. And to do that, we first need to plate our serial dilutions. So when we think of a kombucha mixture, there's both bacteria and yeast. And we need plates that have the nutrients that the bacteria need to grow, which we use these LB plates. LB stands for lysogeny broth plates. And then we have YPD plates, which are what yeast prefer to grow on. And YPD stands for yeast peptone dextrose. And so they're similarly composed, but slightly different. The next step is to add a few glass beads to our Petri dish and shake it. The purpose of using the glass beads is to ensure that whatever cells are plated on here are evenly distributed across the surface of the plate. And this makes it possible for single cells to pop up and us to count single colonies. And so what I'm gonna do is take my 50 microliters. So I need to change this back to 50. I'm just gonna dot a little bit onto the plate. I'm not gonna touch the plate with the tip though, because you don't wanna like make a hole in it. And then I'm gonna move it off to the side because now I know that I've plated it. So you wanna do this for about like 30 seconds or so till the plates are dry. My arms hurt. Okay, that's how you know you're done.
And now we're just gonna repeat that.
Yes.
Okay.
This is reminding me of my grad school days. I would do like literally hundreds of plates at a time. Hundreds. The last step before taking our plates to the growth room is to make sure that they don't dry out using paraffin. Okay, done. Now we're gonna take our plates to the growth room or incubator.